Good afternoon, everyone. So I put up my title of my thesis actually here is the mannosylated lipo-peptides as model vaccines for targeting the mannose receptor. So I'm going to talk about the synthesis of these vaccine constructs, and then I'm going to talk about the in vitro uptake and confocal studies of these vaccine constructs. And then I'm going to show you some animal studies. And then I'm going to show you the receptor affinity measurement with biocore studies for these constructs. So mannose receptor is a frontline receptor in immune responses. It's one of the first receptors involved in the recognition of the virus, bacteria, or fungi when they enter the body. So it's important to look into this receptor for the uptake of vaccine constructs. This receptor has unique properties. It has different domains on its structure. It has 8 carbohydrate recognition domains, which mostly recognize the mannose on the structures, especially the carbohydrate recognition domain number 4. And it has also some other parts that has already been studied for their binding ability. So this receptor, as I said, has unique properties, and there has been speculations about the conformation of this receptor on the surface of the macrophages and dendritic cells. And there's little known how this receptor is conformed on the surface of the dendritic cells and macrophages, and there has been different hypotheses to describe the binding towards this receptor. But what is known is that this receptor mostly recognizes mannose in the structure of the cell wall of bacteria or viruses or whatever, and then it is binding in a calcium-dependent way. So here is the pathway that mannose receptor is involved in the immunity system. So you can see that the pathogen that is entered into the body is recognized by this receptor, internalized into the dendritic cell, and then presented by MHC1 and 2 to activate T cell and give a CD4 or CD8 response depending on the antigen that is presented to it. So we decided to design and make vaccine structures that contain mannose on their structure and also lipid moiety with self-adjuvanting properties, and also OVA CD4 as a model antigen. This vaccine construct had FAM as a fluorescent tag to be later looked into the uptake of these structures, and also they had some different number of spacers between their mannose moiety to look into the binding of the these structures with the mannose receptor. So I started making these vaccine constructs, and because they were quite big constructs, we had to synthesize the whole construct into 2 parts. First, I synthesized the mannose containing part with the fluorescent tag attached to it, and then I synthesized the part with 2 lipid moieties and then OVA CD4, and then clicked them together to attach them together. So here is the first part that I had to synthesize the mannosylated building blocks to put into the mannosylated part of the peptide. And the property of this part of the peptide is that unlike other vaccine constructs that are usually clicked into the whole constructs, these ones are put into the peptide linearly, and they're all linked attached. So it's quite similar to what is presented on the cell wall of the bacteria or fungi. And this is the other part of the peptide that has the OVA CD4 onto it. And then I click them together, and here's the final construct with different number of spacers between the mannose moieties. So here's the final library of compounds. You can see I have the main library of vaccine constructs with different number of spacers between the mannose moiety. And then I have some controls without any mannose on its structure or without any lipid in the structure, or the one without any mannose and lipids, to have a look into the different properties of these structures. I characterized these structures with size measurement and TEM, and they showed to form nanoparticles in the PBS, and the size varied between 100 to 200, and the TEM size was slightly smaller due to the dry out on the TEM disk. So I did an uptake study with dendritic cells and macrophages from splenocytes, because they are a good source of dendritic cells and macrophages. And if you look at these graphs, they show you that in the dendritic cells, you can see that the uptake was quite high for the 3 main constructs, but also quite high for the one that has lipid only on its structure. And I did some mannan inhibition assay. So mannan is known to be a ligand for the mannose receptor, and it binds to the mannose receptor. So what we did was to pre-incubate the cells with mannan and to stop the constructs from being uptaken, and then add the constructs to see how much the uptake is receptor mediated. So we saw some reduction in the uptake. As you can see, this difference shows the amount that is receptor-mediated uptake. And then I did confocal imaging to show that these constructs are being uptaken to the macrophages. And here is the one that I did for control, that I did for mannan added with construct, and you can see the difference between these two. So we wanted to have a look at the effector T-cell response of these constructs, and we found out that when we studied them with the OT2 cells in vitro, we saw that construct 1 and 3 and 4, the 3 of them have quite significant proliferation ability on the OT2 cells. And then I moved on to the animal study that was using OT2 mice as a model mice that has T-cell receptor on its T-cells, and we purified the cells and injected into the black 6 mice, and then immunized and looked into the cytokine release and activity. So the pathways that here we're looking at are the T helper 1 and T helper 2 pathways. Also, we had a look at the CD8 response. Although my constructs have CD4 epitope on their antigenic part. So the proliferation of CD4 showed that construct 2 and construct 4 and also construct 6 had proliferation ability on the CD4, and CD8 was construct 2 as well. So we moved into intracellular staining assay, and here you can see that construct 2 had some interferon gamma response, and construct 4 had IO2 response, and not much significance in the other constructs was observed. Here is a cytokine beta array study, and it's a heat presented in a heat map table. You can see that IO2 production for constructs 1 and 2, and then IO4 and 5 production in constructs 1 and 2 as well. And construct 4 also gave IO2 and IO5 and 6. So to me, it seems like these constructs with both lipid and sugar on them, except for number 3, they were able to activate both T helper 1 and T helper 2 pathway to some extent. And construct 4 seems to be significantly doing only T helper 1 activity, which has only lipid on its structure and is kind of predictable due to cell-like receptor activity. So I moved on to biocore studies. With this study, I wanted to show the actual binding affinity to the mannose receptor. With BioCore, I showed the molecular interaction of these vaccine constructs to the mannose receptor. BioCore basically is a system that contains a microchip that you can load onto the receptor or whatever you want to measure, protein, moiety, onto the surface of this chip, and then pass through the binding compound in a flow cell to measure the binding affinity due to changes in the light reflection, which gives you a signal to have a look at. So a normal signal in the BioCore system consists of injection, which is the sensogram normal starting part, and then there's association, and then there's dissociation, and then regeneration. So we want to have a kind of sensogram that looks like this. So I started with kinetics for Mannan, and Mannan showed quite nice binding properties for these receptor loaded onto the flow cells. So here you can see I used different concentrations of Mannan, and I studied the kinetics, and you can see the binding is slow, and the dissociation part is also very slow. So the results that I observed that for example, group 1 of the peptides, which has 0 space, there was a nice kinetics when the response unit went up to the 25-30 when I looked at it. And then for the group 2, the response unit went to 20. And there were differences in the kinetics when you compare these vaccine constructs together. And group 3 also showed some high response units. And this is group 4, group 5, and group 6. So you can see that group 5 doesn't have any lipid moiety, and it also didn't bind that hugely. And this group that didn't have any lipid or any sugar also didn't bind much. So if we want to just look at one concentration of these binding response units, you can see that group 1 gave us best response unit with this 25 micromolar concentration. And group 2 was kind of less, and group 3 and 4 were better, which these data kind of matches the OT2 proliferation assay that we observed. And Mannan, of course, was the best, which is our control group. So in conclusion, I made the library of mannosylated lipo-peptides with fluorescent tag that were successfully synthesized and purified. And we also designed mannosylated lipo-peptides that had better uptake when compared to the control library. And most of the uptake was receptor mediated, and for cytokine release, group 1 to 3 showed activation of T helper 1 and 2 pathway. Group 4 showed activation of T helper pathway, T helper 1 pathway. And group 1 and 3 were involved in the uptake. I'm discussing this, that is probably through mannose receptor, and these ones showed best binding affinity when I did the in vitro studies. And group 3 showed the most selectively toward T helper 2 response with and no IL-2 response. And BioCore studies confer an affinity of constructs 1 to 4 to the mannose receptor. Thank you very much.
Any questions? Shirani?
You are using a CD4 kit, and you are saying that you are getting CD8 response.
So there have been some speculations that there is a hidden B cell epitope inside this sequence of OVA, but we kind of didn't see much activation. Yes. Anyway, we looked into it. There might be some cross-presentation inside the cell, so due to presence of the mannose. Because mannose receptor, there has been papers that they discuss when the uptake is receptor mediated, then there can be some cross-presentation in the pathways involved in the afterwards.
Do you have the size of the peptide that you can add? Is there a size you can add, or is there a maximum size?
For the biocore studies, as long
you are using, I know that you are using all antigen, but you are using a true vaccine context.
Are you talking about the binding affinity part?
Just like a protein that you're going to peptide that you're going to
Okay, so there has been some studies that showed they have shown that small as 9 amino acids they get some immune responses. So is that what you? I don't know about the maximum, but I think as long as I know, the bigger is better, because the size when the size is bigger, probably there is some adjacency properties involved in the size. So I'm not sure, but for my constructs, the 3 main library of constructs, they had bigger size when compared to the control library, and they gave better affinity to the receptor, they gave better immune responses. So,
Are there any other questions? Yes.
Just a technical question. When you're assembling these molecules in your synthesis, how do you confirm that the final structure is the one that's drafted?
so there is mass spectrometry, and there is some softwares that you can draw these amino acids next to each other. The same software that I've used to show you the structure. This software has ability to predict the molecular weight of the final construct. So you look at this molecular weight in the mass spectrometry, and with HPLC, that one is purification, that you take the pure fraction and put it in the mass spectrometry to confirm that this is the exact structure that you're looking at. Thank you.
Do you do any flow cytometry work to see whether you're inducing multi-active T cells secreting multiple cytokines? Because your ELISA will only tell you the total.
So with ELISA, I didn't see any antibody response.
No, I'm talking about cytokine production. So did you use any flow-based assays to see if you've got cells secreting more than one cytokine, say gamma interferon and TNF, in any of your constructs?
So unfortunately, there was a human error with the interferon gamma. We use these kits to do flow cytometry for the ICS assay and the CBA assay. For the interferon gamma with the CBA assay, we misplaced one of the dyes, and then we didn't see the interferon gamma response. But for other than that, everything else was there. So.
Any other questions? Okay, if you could join me in